Okay, I would like, this is research that I'm going to describe, applied neuroanatomy, linking between spatial scales and the visual system. This is research that we've done over the last 5 years in my laboratory at Stanford University. It was spearheaded by Kevin Weiner, sitting over there, and Mona Rosenka and Jesse Gomez are sitting over here, as well as Michael Burnett and Anthony Stigliani. I would like to thank our collaborators in the labs of Professor Zillis and Katrine Ametz. So our lab studies the visual ventral pathway, and this is a hierarchically organized pathway starting from V1 in the occipital lobe and organized through a series of retinotopic map going into ventral temporal cortex. In addition to retinotopic maps, a high-level visual ventral temporal cortex contains high-level visual areas processing information for particular domains, including face-selective regions, place-selective regions, body, objects, and word-selective regions. In this presentation, I will address two big picture questions. First, is there a structural functional coupling in the human ventral visual stream? And if so, what is the spatial scale of this coupling? Second, if we find such a coupling, perhaps we can gain some insights into the anatomical constraint that underlies the functional organization of the human ventral stream. So there's a large body of research suggesting that in primary visual cortex, or V1, there's a very strong coupling between function and structure. For example, in every participant's brain, V1 is always within the same macroanatomical structure, the calcarine sulcus. Not only V1 is found in the same part of cortex, but there is a very strong relationship between visual field representation and macroanatomy. For example, the polar angle representation in V1 goes from the lower field representation in the upper lip of the calcarine to the horizontal representation in the fundus of the calcarine to the upper visual field representation in the lower lip of the calcarine. Likewise, there's also systematic representation of eccentricity, which progresses from posterior to anterior, from foveal to peripheral representation. And in fact, this coupling is so reproducible across participants that Noah Benson and his colleagues have developed a mathematical algorithm that just from the cortical folding, you can predict the entire visual field maps of 3 visual areas, V1, V2, and V3. So we ask, is there a similar functional structure relationship in high-level visual areas of the human ventral stream? So several researchers, including prominent researchers, have noted that there might be impediments to functional structural coupling between early sensory regions. And this comes from 2 sources of variability, anatomical and functional variability. For example, in a paper written by Van Essen and colleagues in 2012, they write, the complexity and variability of cortical convolution are an impediment to visualizing results and making accurate comparisons across individuals. In the same papers, they've also noted about functional variability across individuals. For example, they write, another confound is that the size and shape of each cortical area can vary by 2-fold or more across individuals. So because of this structural and functional variability, the prevailing view suggests that both sources of variability might contribute to a lack of a clear functional structural coupling outside of early sensory cortex. However, I will argue that with improved measurements, we could find significant structural functional coupling outside early visual cortex. And these improvements come from several sources. First, our research uses single subject analysis. We do multiple measurements in the same participants. As noted by Reiner previously, there's benefits to using higher resolution fMRI, improving the accuracy and precision of measurements. We do not spatially smooth the data. We use cortical surface visualization. And all of these together improve our measurements and enable us to see these couplings. And also, I will talk about macroanatomical structures, and especially tertiary macroanatomical structures. As a model system, I will talk about 2 regions in the ventral stream. One are phase-selective regions, shown here in red. These can be found in every individual subject's brain using a short fMRI scan, and they're characterized by higher responses to phases compared to other stimuli. Another set of regions I'm going to describe today are place-selective regions. Again, they can be found with the same localizer scan, and their characteristics is significantly higher responses to places compared to other stimuli. So let's start with looking at place-selective regions. So this slide shows 6 medial views of 6 individual brains. The place-selective region is shown here in green, and the collateral sulcus is shown here in blue. And these is an inflated view where gyri are indicated with a lighter gray than the sulci. So you can note that in every individual example, this place-selective regions overlaps the collateral sulcus. So even though this region is normally referred to the parahippocampal place area, in fact, it's a different structure, the collateral sulcus, so we refer to it as the collateral sulcus places. Another thing that you could note is that the collateral sulcus is actually quite long. It spans from the occipital lobe to the temporal lobe, and in fact, it's more than 5 centimeters in length. So having an anatomical structure in the name doesn't actually give you perhaps the desired anatomical specificity. Nonetheless, you can see that it actually doesn't seem so variable in its location across these individual participants, so perhaps there is more to functional structural coupling. So in order to look at this more deeply, I'll just show you first one individual brain and highlight some important anatomical features. So again, I'm highlighting the collateral sulcus with this dashed blue line. Superior to the collateral sulcus is an anatomical structure called the lingual gyrus, which is shaded here. And the lingual gyrus contains 2 sulci, the posterior lingual sulcus, PLS, and the anterior lingual sulcus, the ALS. In about half of the brains, the ALS intersects with the collateral sulcus. And if we superimpose on this anatomical image the location of place selectivity, you can see that the place selective activation seems to overlap this intersection between the ALS and collateral sulcus. So to evaluate the anatomical predictability of place selectivity, we use Cortex-based alignment to generate a probabilistic ROI, and then tested if this probabilistic ROI could actually predict place selectivity in new brains. So we generated a probabilistic ROI from 12 subjects and projected it to the free surfer average brain. And one thing that you can note that this probabilistic ROI seems to have the same anatomical features as individual subjects, because you can see that the probabilistic place selectivity seems to happen at the same place at the intersection between the ALS and the collateral sulcus. If you also look at the location of our probabilistic ROI, shown here in the contour, compared to an unstressed map of place selectivity using 24 other subjects, you can see that the locus of highest place selectivity within the ventral stream is within our probabilistic ROI. And this result was replicated with 2 independent data sets, additional data sets, one from Nasser et al. And one from 247 subjects from the Connectome project. So the question is, can we also predict the place selectivity in an individual subject's brain from this probabilistic ROI? So what we do is we do an inverse Cortex-based alignment, now aligning this ROI into individual subject's brain, and then extracting the signals from individual subject brain, and then we can test if it actually predicts place selectivity in each individual subject's brain. And this is what we did in the third analysis, and this is basically extracting the activity from a different experiment in 12 independent subjects, and you can see that in these subjects, the highest responses from this region are for indoor and outdoor scenes, pictures of places, and they're significantly higher than responses to other stimuli, such as objects, textures, or faces. So this is an example where using the structural functional coupling, we're able to predict place selectivity in new brains. This ROI is published. You can, it's on the free surfer average brain, and you can freely download it from this link. So this is one example of structural functional coupling in the ventral stream. Another anatomical structure of interest that I'd like to point your attention is a mid-fusiform sulcus. So this is now a ventral view of the brain. And in this ventral view, I'm going to highlight a gyrus. That is one of my favorite gyri in the brain, the fusiform gyrus. And this is particularly because it's a human-specific gyrus. Within the fusiform, there is a small sulcus called the mid-fusiform sulcus, illustrated here in red, and it bisects the fusiform into a lateral and medial fusiform partitions. Even though we have found it in all 158 hemispheres of participants ranging from the age of 5 to 85, you will not find it in typical atlases that are used in neuroimaging, like the Duvernoy or Talirac atlas. And I think this is because it's tertiary sulcus, and if you have only one brain that you're building your atlas, you might not be confident about how reproducible it is across participant. Nevertheless, if we look at many brains, we become very confident that the sulcus is there in every single subject's brain. Its primary characteristic is that it's half as deep as adjacent sulci, the collateral sulcus, and the occipital temporal sulcus. So one identifying feature of the sulcus that if you look at a coronal slice, it generates this omega shape together with the collateral and occipital temporal sulcus. So why am I showing you the mid-fusiform sulcus? I'm showing you because we noticed this first because we looked at phase selective activations. So these are 3 ventral views of 3 independent participants where the phase selective activities are indicated here in red. And typically in each subject brain, we find 2 patches in the fusiform gyrus, 1 patch that overlaps the mid-fusiform sulcus, the MFS, we call it M-fuse faces. A second patch that's also on the lateral fusiform, but it's 1.5 centimeters more posterior. We call it P-fuse faces. Often people lump them together to call them the FFA. And then we have a third patch that's outside the fusiform gyrus. It's in the inferior occipital gyrus, and we call it the IOG faces. And you can note that in these 3 individual examples, there seems to be an alignment between the anterior face patch with anterior tip of the MFS and the posterior face patch and the posterior tip of the MFS. And again, we wanted to test how reliable the MFS is for predicting functional activation, and this time phase selective activations. So what we did, we took 14 independent brains. We identified based on their anatomy, the MFS in each brain, and then we placed a 1 centimeter disc on the lateral fusiform aligned either to the anterior or posterior fusiform gyrus. Placing a disc in the anterior, next to the anterior MFS predicts about 83% of M-fuse faces. Placing a disc on the posterior MFS predicts about 50% of P-fuse faces. If you're placing disc according to Talirac coordinates, you're going to do very poorly. You're only predicting 18% of the voxels. You might also note that our predictability is better in the anterior end than the posterior end of the MFS, and this is because this is a more stable anatomical landmark across subjects. The anterior MFS, anterior tip aligns with the posterior end of the hippocampus, but there's variability across subjects in the lengths of the MFS. Another thing that you could look at in the ventral stream is looking at large-scale maps. For example, this is a large-scale map of eccentricity bias shown in 3 individuals, a child, an adolescent, and an adult. The hot colors indicate regions that prefer foveal compared to peripheral stimulation, and the cold colors indicate regions that prefer peripheral compared to foveal stimulation. And you note that in each of these 3 examples, there's this lateral medial separation between fovea and periphery. And an interesting feature is that if you now draw the MFS in each of these individuals, you can see that the MFS seems to be a good predictor of this lateral medial gradient between foveal and peripheral representations. And we actually measured what is the distance between the functional boundary and the anatomical boundary, and independent of the age group of the participant, the gradient in the lateral medial eccentricity map is on average 4 millimeters from the fundus of the MFS. So back to my first question. 1, is there a structural functional coupling in the ventral temporal cortex? And the answer is yes, and the scale of this coupling is quite fine. It's less than 5 millimeters. And I've shown you 2 examples where macroanatomical landmarks can predict function. One is an example of the intersection between the ALS and the collateral sulcus that predicts the most probable location of the place selectivity in the ventral stream. And the second is an example of the mid-fusiform sulcus that predicts phase selective regions as well as transition in this eccentricity bias map. This brings us to the second question of what are the anatomical constraints that underlie this functional organization of the human ventral stream. And we hypothesize that two factors might constrain this organization. One is long-range white matter connections, and the second is a cytoarchitectonic structure that might constrain this function. So I'll show you an example of fiber tracts. And here again, pictures of 3 individual subjects. And I'm showing you one set of fiber tracts here in red, and these are fiber tracts that connect between phase selective regions and the anterior end of the temporal lobe, and in green are fiber tracts that connect the place selective region and the anterior end of the temporal lobe. And you can note that in each of these examples, these fibers are largely parallel, and in fact, they don't intersect. There's about 3% intersection in the back, and only less than 14% intersection in the front. So these are largely parallel tracts that segregate information from place and phase selective cortex. Interestingly, the endpoints of these tracks, if you look at them relative to the MFS, is also consistent across participants, whereas the endpoints of the tracks associated with phase selective cortex really align quite nicely with the MFS, whereas the endpoints of the track associated with place selective cortex are 1 centimeter more medially. So there seems to be also a coupling between connectivity and macroanatomy. A second example is from cytoarchitecture. Using the method that Nicola described previously, Julian Casper and Simon Lawrence from the Zillis and Amunds labs have described 4 cytoarchitectonic regions of the Fusiform Gyrus and neighboring sulci, FG1, FG2, FG3, and FG4. And visualizing the location of the MFS on this example postmortem brain now shows that the MFS seems to predict lateral medial transition in cytoarchitectonic maps between FG1 and FG2, as well as between FG3 and FG4. And we did the same exercising of measuring the boundary between cytoarchitectonic regions relative to the macroanatomy, and we find that the boundary between FG1 and FG2 is within 5 millimeters from the MFS, and the boundary between FG3 and FG4 is within 2 millimeters of the MFS. And again, this structural cytoarchitectonic coupling is better at the anterior end of the MFS because this is a more stable macroanatomical landmark. So what does this mean that the MFS predicts cytoarchitectonic boundaries? This means that in different sides of the MFS, you will find cells with different cytoarchitectonic structure. For example, FG2, which is lateral to the MFS, has a denser cytoarchitectonic structure compared to FG1, which is medial to the MFS. And if you remember the lateral medial separation of the eccentricity bias map, this might be because of differences between foveal and peripheral computations that might require cells of a different structure. And another thing that you might wonder that since we've shown examples that the MFS predicts both functional and cytoarchitectonic transitions, perhaps there is a relationship between a cytoarchitectonic area and the functional regions defined with fMRI. Now, evaluating this possibility necessitates relating cytoarchitectonic data in histological slices in postmortem brains to functional regions in living subjects. So together with the Zillis and Amunds lab, we had to develop a pipeline that could link these very different kinds of data. So what we do in this pipeline is we take the cytoarchitectonics that have been defined at ULIC. They also have anatomical MRIs of the same postmortem brains before they have been sliced, and they have transformed them to the MRIs of each individual. What we then do, we take these MRIs and generate cortex-based visualization of each of the 10 postmortem brain, and then what we can do is generate a probabilistic map of a cytoarchitectonic region on an atlas like the Free Surfer atlas. And in fact, we've created such a cross-validated atlas of the 8 cytoarchitectonic regions of the ventral stream. As this atlas is available both in Free Surfer and Brain Voyager formats. You should feel free again to download it from this link. And if you want to hear more about how we generate this atlas and how you could use this, please visit Mona's poster on Tuesday. So now that we have this cross-validated atlas of cytoarchitectonic region in the ventral stream, what we could do is use cortex-based alignment and project the probabilistic map of each cytoarchitectonic region into individual living subject's brain, where we have defined functional regions, and see if there's an overlap between a functional region and a cytoarchitectonic area. So let me show you the results of this analysis. First, I'm going to show you the group map. So the shaded region shows the probabilistic map of the cytoarchitectonic area FG4, and the contour shows the probabilistic location of our phase-selective region, M-fuse faces, and you can see that at the group level, M-fuse faces is largely constrained within cytoarchitectonic region FG4. We also projected it to an individual subject's brain. So here, for example, individual subject's brain, again, the colored region shows a probabilistic map of FG4, and the contour now shows the individual subject's M-fuse faces. And we quantified in each of our participants how much of their M-fuse phases is within each of the cytoarchitectonic region. And you can see that the only significant region that we found was FG4. And across our participants, 83% of M-fuse faces lies within FG4. So this suggests a structural functional coupling between functional regions and cytoarchitecture in the ventral stream. If you look now at place-selective cortex, we also find a structural functional coupling, but this is now between a different cytoarchitectonic region. And you can see both in the group map as well as in the individual subject brain that there seems to be a coupling between the collateral places ROI and a different cytoarchitectonic region, this time FG3. And across our participants, 72% of collateral sulcus places is found within FG3, and there's no significant portions of them in other cytoarchitectonic areas of the fusiform. So what does this mean? This means that face and place selective cortex are cytoarchitectonic different. So this is very exciting because now we can actually project back our functional regions all the way back to the cytoarchitectonic slices, and for the first time actually visualize the cytoarchitectonic structure of functional regions. So during the reverse process, we take the functional MRIs, project them back to cytoarchitectonic slices, and then we can extract the cytoarchitectonic profile from face and place selective cortex. And if you look at the gray level index of face selective cortex compared to place selective cortex, you can see that they have different cytoarchitectonic structures. For example, face selective cortex has a prominent layer 3 and a layer 4 with intermediate and large pyramidal cells. But if you look at place selective cortex, it has a dense but prominent layer 2 and layer 4 with granular cell. And so this is really important. And I think this is really important because those of us who use fMRI, we're measuring indirect measures of brain function. We've studied these regions of face and place selective regions based on their functional characteristics for now over 20 years. But this really suggests that there might be very different neural hardware that supports these computation, and it provides some direct cellular level evidence for functional specialization in the human brain. Okay, so just so you won't think that life is so simple, I would like to put one caveat. So I already said this. I'm going the wrong direction. Why is it going? Okay. The caveat that I would like to say is that I'm not trying to say that there is a one-to-one relationship between function and cytoarchitecture. So face and place selective cortex are cytoarchitectonically distinct. But if you look at additional functional regions within the ventral stream, you'll first find that the cytoarchitectonic areas are bigger than the functional regions, and you could also find within a cytoarchitectonic area more than one functional regions. For example, within FG4, you can find face, body, and word selective regions. And if you look within cytoarchitectonic area FG2, you can also find face and word selective regions. So this suggests that there's a one-to-many mapping between cytoarchitecture and function. So to end, I would like to propose a new hypothesis that there might be two computational axes in the human ventral temporal cortex. One is a lateral medial axis that we call it the axis of specialization because it enables processing of information from different domains as segregated cortical regions, which can be done either in different cytoarchitectonic areas like FG4 and FG3 for face and places, or within the same cytoarchitectonic region, for example, faces, words, and bodies. And we think that this lateral, why some regions are in the same cytoarchitectonic area has to do with foveal versus peripheral computation. So face and word processing use foveal vision, whereas place processing use peripheral vision. Across the ventral stream, it's known that fovea, cells processing foveal information differ in their structure than peripheral information, and we think that if you have regions within the same cytoarchitectonic area, they might have similar foveal or peripheral biases. The second axis is the anterior-posterior axis, and we think we would like to call it the axis of transformation. One thing that we know about the ventral stream is that the hierarchy proceeds from back to front, and a feature of hierarchical processing is increased size of receptive fields. So we hypothesize that within a cytoarchitectonic region, there might be cells of similar receptive fields, and as you move to the next cytoarchitectonic area, maybe the receptive field expand, and this could even lead to having regions of the same domain, but in different cytoarchitectonic areas because they might have different receptive field sizes. So to conclude, is there a structural functional coupling in the human ventral stream? Yes, there is, and it's at a spatial scale of less than 5 millimeters. What are the anatomical constraints that underlie this functional organization? We believe that both a long-range white matter connection as well as a cytoarchitecture constrains this functional organization. I will also like to underscore some insights in future directions. First thing is that I hope I have convinced you that understanding the structural functional relationships provides important insight also into computational strategies of the brain. And also, we hope that maybe in the future we could have in vivo measurements that would enable us to do these measurements maybe at a finer scale in vivo with an individual brain, and that might provide increased precision and direct links between structure and function within the same individual brain. So thank you for your attention, and thanks to all the people who worked on this research.
We take questions to Kalanit, and then we open to general discussion, please.
Hi, really wonderful work. It's kind of a general question. Do you think that, so you've shown that one observer-dependent cytoarchitectonic region will map to several functional regions. Do you think that's actually the case, or that that particular measure of structure isn't differentiating these functional regions, but they are actually computationally circuitry different?
So I think both things are probably true. So we have an additional evidence. For example, if you go to Mona's poster on Tuesday, we compared other functional parcellation of the brain, for example, retinotopic maps. And let's say within FG3, we find more than one retinotopic map. We have PHC1 and PHC2. Within FG1, we have 2 retinotopic maps, VO1 and VO2. So I think, and this is different, for example, between V1, because in V1 there is a one-to-one relationship between a cytoarchitectonic area and a functional area. So this is one thing. The second thing is that this is only one feature of anatomy, as you noted, and Carl has just shown a really wonderful talk about receptor architectonic, and it might be that they're finer scale distinction that would actually find also separations between, let's say, face and word selective regions. And I think this is one of the directions that we think is going to be very exciting for future work.
Thank you. Now, other questions for Kalanit? I'd like to ask
There's one more question.
Very nice talk. And I'm wondering that there is some kind of spatial disagreement between the real neuronal activity areas and ephemeral activities. And I wonder how the cytoarchitectonics overlaps well with the bold activity areas.
So I'm not sure I'm understanding your question.
So the bold activity actually occurs in the distal part of the heart area because of the innate property of the bold signal.
Okay.
So there is saying that there is a little bit discrepancy between the real hotspot in terms of neural activity and the bold activity. However, you show that fMRI focus is well overlapped with the cytoarchitectonic map. So how can you explain on that?
So there's some evidence that suggests that there might be a discrepancy between like spiking activity and bold activity, as bold activity is best coupled with LFP. But in the domain of face processing, there's very nice work from Doris Tsao's work that shows that if you look at in the monkey brain at fMRI patches that are selected for faces, and you actually put in electrodes there, you'd find that a very high proportions of neurons are face selective. So I think there is accumulating evidence that some of the fMRI activity is actually very tightly linked to the spiking activity. I think what our data shows that we can actually just try to find out that there are actually some cellular basis for the specialization in the brain. So perhaps computation on faces require different neural architectures and computation on places, and this is why we have dissociable cytoarchitectonic structure for these regions.
Another question?
Thank you very much for the talk. I was wondering if you could speculate on how this correlation is specific for the ventral stream, or if you could expect with your hypothesis of the two different lung connectivity and cytoarchitectonic differences, if it would be the same in the auditory cortex, for example.
So I think that there are probably similar principles across the brain, and I would really like to see a similar kind of work done in other parts of the brain, and we think that the same heuristics would probably be relevant for other sensory areas. So I think auditory cortex would be a really good place to examine. For example, Carl has shown before, and Rainer has shown before, that Heschel's gyrus is very clearly linked to auditory cortex. It has its own cytoarchitectonic structure, its own receptor architectonic. It has very clear columnar structure, very much like visual cortex. So I think it's very likely that we'll find similar principles in other parts of the brain. Okay, thank you.
Thank you. I think we ran out of time. We're late. But there is some time for general discussion. If you want to say what you think about the sessions, or you have burning questions, please go up to the microphone. It's your time. Here we are. Right.
Sorry for delaying the break. This actually was a question for Kalanit, but I thought I'd ask it later. I believe in the first part of your talk, you were mentioning that there is a tight functional structural correlation in terms of curvature. Now, this seems to be a bit discrepant with the recent findings from the Van Essen's group and Matt Glasser's atlas, which show that if you use curvature registration, you actually move more away from the functional match between subjects. Now, of course, that is a full atlas, full brain thing, and your research is mostly on a tight area. I was wondering whether you have any thoughts, or maybe are you planning maybe to use the HCP data set to try to see whether this functional structural coupling exists in those same subjects?
Okay, so we've actually looked at the HCP data in two ways. One is looking at place selectivity. So if you look at the paper, you can see the HCP place selectivity map in our paper. And also we looked at the HCP data in terms of their myelin maps. And I think what's really different between our approach and their approach is actually looking at the tertiary or the small cell site. And they might be better predicting than the big cell site because the big cell site are really big. And this is kind of like the cloud cell is huge. So just maybe if you don't pay attention to the very small details, you're not actually gaining a lot of information. So if you take, let's say, the MFS and compare it to the HCP data, you find that the MFS really predicts the transition in myelin maps in their data. It also, the ALS and the collateral cell intersection also predicts the place selectivity. So I think that one is attention to small cell site that I think it's glossed over. I think there is a need to use multimodal measurements, as was asked in the previous questions. Perhaps if we combine measurements, we can do much better. So I think this is definitely an approach that's worthwhile doing. So I think these are just complementary approaches, and perhaps slowly but steadily we'll figure it out.
Actually, I have a question for you. I think you will see at this HBM there'll be two trends. There'll be neuroimagers. They'll try to collaborate with anatomists. And there'll be neuroimagers that actually don't care at all about neuroanatomy. They'll go solo also. Well, so here's the end of these sessions. Which group do you belong to? The solo or the collaborators with anatomists? Do you think anatomy is going to make a difference to your future research or not? I know you just attended the neuroanatomy session, so you have to. But who thinks that actually neuroanatomy, at the end of the day, I mean, we we can easily analyze our neuroimaging data, you know, with or without considering all these anatomical aspects. I'm referring to graph analysis and that kind of thing, if I have to be more explicit.
I can only get myself in trouble here. I love this work, but I would not put a constraint on myself to say that every move I made in trying to diagnose a child's diagnose a child's reading disability or a major depression had to have this before it was real science. There's still a lot of things you can do, which doesn't in any way diminish the beauty and value of what's going on in this room. I think this room is a fantastic room, but but there's lots of other very valuable things we can do before we figure all these things out. I have a question for all of you, though. Kalanit, you say this. So I should confess that I'm in a room with Kalanit once a week. I've never asked her this question. For the computational insights that you all think that you will arrive at from looking at the cellular structures, this really flies in the face of the entire development of machine learning, where those guys who are ruling the world right about now use a computational structure in which every layer is the same as every other layer apart from the spatial spreading and so forth. So I'm wondering, do you guys feel like you have a play that you're going to say, you know what, the next generation of machine learning actually better pay attention to this?
I just started a course with Dan Yaman, and this is one of the questions that we asked. This is a course between machine learning and neuroanatomy. So I think the brain has been optimized across evolution for many millions, probably, of years, and has optimized us to be very well behaving and reacting to our environment. So I think putting insights of evolution into the neural network has to be a necessary next step. Now, if you just want to classify images in Google and say, if it's your face, somebody else, you couldn't care less. But if you want to understand how the brain works, I think you really have to put these anatomical constraints into deep neural networks, otherwise they won't explain the brain. So the fact that there's cluster activation, the fact that there's different cellular structure has to be explained by these neural networks. So one of the things that you could do, for example, build a neural network that has no spatial structure, and there are many of them built right now, or put the neural network, it has some structure, and see, does it give you any benefit? And that would be a way to use these neural networks to get some kind of, to glean some computational insights.
There is another example. If you have an area which you have characterized by functional neuroimaging, and you find the same area, for instance, in the monkey. You have characterized it by the same tasks, and you would say, well, these are comparable areas. And then you do a receptor study of these areas in monkeys and in human, and you find considerable different receptor expression in the monkey. I have shown only an example where the receptor expression is identical, but I can also show areas, especially in the so-called higher cortices, multimodal association cortices, where humans have a receptor expression which is completely different from macaque monkeys, vervet monkeys, New World monkeys. How can you explain this by the present status of simple machine learning aspects? It puts everything together, but it's different, and we should really look which sub-function, subspeciality of function is supported by this tiny but significant receptor difference between a monkey and a human brain.
Karan. Thanks. I also want to add to the question of Brian. I would say they have already learned from the brain, because when they move from fully connected network to convolutional network, they already stolen a lot of the knowledge we have from the brain, that we have small receptor fields which locally filter the environment, the visual input, especially for the visual, of course. And then, so the architecture which has been changed led then to the high accuracies on the object recognition tasks, and that boomed then this kind of networks. And I think it will be happening like this. I mean, machine learning is still considered mainly as artificial intelligence. So I would say they're very smart, these people, and they will use everything, and now comes an important point, which ultimately will improve the performance of their goals. So that means it's a nice test. I'm looking forward, very curious about this, because I will see what changes or what structures in the brain are there because they have helped to improve human performance, like Kalanit says, to performance and make us so powerful most of the time. And on the other hand, which things are there just by coincidence? So is a certain partitioning helpful, really helpful for improving performance, you know, in all the wide tasks, not just one, but like human-like performance, right? And ultimately, it's like another selection, another Darwinism, which will kind of tell us researchers what of the brain's partitioning from molecular, neuronal, macrostructure, mesostructure, what of these things are really helpful to better performance, and which are just there because of anatomical constraints, things which were there, built on top of each other, might not have led finally to the most elegant and powerful final structure. So it's a very interesting test, which, in my opinion, these people do now, to see what in the brain is very important. Like convolutional ideas are very important. They have shown this already in the visual domain.
So when I was listening to your talk, Carl, I was wondering, you have shown these receptor fingerprints, and in the context of these computational networks, where it's a balance, as Jesse mentioned, between excitatory and inhibitory. So do you think you could use that into a computational network to predict like how much excitation, the balance between excitation and inhibition in different regions in the network, or maybe?
Yes, I think so, but the data show me that also the modulatory receptors play an important role. For instance, when you look to the muscarinic M2 receptor, which is a modulatory receptor, it is very, very high primary sensory areas. They stand out. Or otherwise, the general rule that the supragranular layers have higher receptor densities, overall receptors, than the granular layers. This rule is broken by the nicotinic alpha-4 beta-2 receptor, which has a clear peak in the granular layer of primary sensory areas. So I would very much like to use this data in computational modeling. But presently, we made already 2 attempts to get some ideas how to model this. We failed up to now. But this does not mean anything. This means only that we are not good enough in computational modeling. We have to learn.
Okay, but I think we're coming to the conclusions. Any other questions? Yes, please go up to the microphone and speak. Oh, it's the microphone. There's another one down there, so I can kind of...
So I was wondering for the networks that, for example, the default mode network where you found the receptor fingerprints, basically. Do you think that also the cortical architecture can predict from what areas are in what networks, for example, or do you think that it is specific for the fingerprint of the receptors?
Thank you. That's really interesting. We did hierarchical cluster analysis of all these areas, and in the hierarchical cluster analysis, it does not show up this distinction between default areas and non-default areas. But in this case, the analysis was mainly done for all layers. So we have to go into more detail to see what's going on there. But if we study the layers, as I have shown here, we have a clear separation between the default areas. But what puzzles me really is that the excitatory umpire receptor and the density of this receptor is negatively correlated with the glucose consumption, with the glucose consumption. I thought it should be positively correlated. It's positively correlated in all the other areas. So in the primary sensory areas, in other multimodal association areas, but not there. So this may be an interesting mechanism, a molecular basis of a mechanism which we still do not completely understand.
Okay, thank you.
Carl, now that you're unlocking the secret of the receptors of the default mode network, tell us what chemical we can use to inhibit the default mode network so that we can increase our concentrations when we do science or write up papers. Tell us.
I will do a multi-component analysis, and write you an email in 2 weeks, and you have to promise not to tell somebody else what is the secret. I don't know, but we can really separate via multidimensional scaling analysis or multivariate analysis, which receptors contribute most to this distinction. And my prediction is that it's again not a single receptor. It's the right mixture of different receptors which make this distinction.
Okay, I think we can one more. That's an interesting discussion. By the way, the reception's at 7:30, so we've got plenty of time.
You asked how to turn off the default mode network to increase your concentration?
Correct.
Okay, you should know that when you turn the default mode network off, you go to sleep or you become unconscious. So,
right, that's okay. That's interesting.
So you asked, you want...
Not completely off. What I mean is it's kind of a tend more to external sensory...
Okay, we can do an experiment, but I think if you try it, your ability to concentrate will go down.
Right, okay. But what about ADHD and people that have concentration problems?
Well...
that's a mind-wandering kind of problem.
Yeah, they take, basically speed, or modern versions of speed.
It works.
Yeah, it does work. But
Okay.
that's, noradrenergic, not... you want to be awake.
Dopaminergic.
Yeah, dopaminergic. Yeah, you want to upregulate your muscarinic receptors and your dopaminergic, and maybe turn down your 5-HT. I don't know.
That's it in a nutshell. That's for you, Carl. That's a suggestion. Okay, so I think we should conclude these sessions. It's very interesting. The substance that you can use, which is receptor specific, it will knock down all your receptors, is alcohol. But drink in moderation this evening, and I look forward to this, to talk to you about different substances to stimulate your concentration. Thank you very much. Thank you to the